So let me introduce our next speaker, and I asked how to pronounce it correctly. It's Srikanth Ramaswamy, and he's the director of the Neural Circuits Laboratory at Newcastle University, and he's going to talk about neuromodulation. So yesterday at Max's workshop, those who attended, we talked a lot about, or Max talked a lot, and we listened and learned about neuromodulation. This is another dimension in shaping, and another dimension through which our biological neural networks can adapt. But I think we're going to hear a lot more about that. So without further ado, please.
Is this mic working? Yeah. Okay. So thank you very much for having me here, and yeah, it's an exciting conference, and hope to learn lots from being here. So I don't know what aspect of neuromodulation you learned about yesterday, perhaps in the clinical sense. So what I'll try to tell you briefly today is how certain signaling chemicals called neuromodulators, how they're organized in the brain, what can we learn about their organization, how do they enable biological neural networks to learn, and how can we mimic some of these principles in biological neural networks into artificial neural networks. So my research combines multidisciplinary, experimental, and computational neuroscience approaches. Really the interface of neuroscience and AI, this evolving interface called neuro-AI, which, as we all know, won the Nobel Prize in Physics and Chemistry this year. So this is a very emerging field, and the synergy between neuroscience and AI can help each of these disciplines learn a lot, to grow synergistically, to evolve together. So what I do in my lab is to study how neuromodulatory systems like acetylcholine or dopamine or serotonin, such signaling chemicals, how are they organized across mouse, monkey, and human brains? So from the previous talk, you learned a bit about brain slices, about patch clamp recordings. So what we do in my lab is to make brain slices from mouse, monkey, and on a lucky day, also from resected human brain tissue, and try and understand how neuromodulatory systems impact cells, synapses, and microcircuits across species. So we're very interested in understanding how neuromodulators capitalize across growing biological network complexity to bring about higher order cognitive functions like learning and memory and attention and arousal. So some questions we're addressing are, how are neuromodulators organized across different mammalian species? Are they the same? Are there differences? And how do they scale with increased biological complexity? And how can we use all this biological knowledge? How can we integrate this knowledge into computational models of artificial neural networks to try and predict how networks learn, how networks evolve, how networks compute? So this is a fairly contemporary picture of modern neuroscience. So what we see here are cavemen and women, right? They're all doing their own little thing without knowing they're all doing the same thing. Perhaps they're studying the effects of neuromodulators in some part of the brain, right? So what if, we could somehow magically bring together what these cavemen and women are doing into some kind of a framework here? So some years ago, as part of a huge moonshot initiative called the Blue Brain Project that started in Switzerland, we set out to map biological organization in a little piece of the brain called the neocortex. This is so we started by measuring how certain components called ion channels, neuron morphologies, their firing patterns, their connectivity, how all this comes together to shape the dynamics of a prototypical neocortical microcircuit, a biological network, right? And we started by gathering data across these different levels of organization, sparse biological data, identified rules and principles of organization, And these rules and principles then constrained algorithms that were used to build computational models across each of these levels. And so we ended up with component computational models that were then integrated to give a complex model of the neocortex. This is quite a dense model, but we are still talking about a piece of brain tissue that's roughly the size of a pinhead, about 0.3 millimeter cube, and yet this small, microscopic, granular piece of cortical tissue contains about 30,000 neurons with millions of synapses. So it's quite a complex Wild West out there, right? So in the brain, state specification and so-called brain states arise partly from these different neuromodulatory systems. So these colored ellipsoids here represent different neuromodulatory systems. For example, blue represents acetylcholine that's released mostly from the basal forebrain and that projects to the cerebral cortex, or dopamine that's released from different sites in the subcortical brain regions and also projects to the cortex and elsewhere in the brain, or even serotonin that's released from a little part of the midbrain called the dorsal raphe, or noradrenaline that's released from locus coeruleus. So all these neuromodulators are released from subcortical regions, and they project to many parts of the brain, mainly to the neocortex, to try and bring about these state transitions and changes that shape behavioral states and higher order cognition. So of course, many of these neuromodulators have been linked to specific functions depending on the target area they project to. For example, serotonin has been traditionally linked with threat assessment. Acetylcholine has been linked to attention and arousal. Noradrenaline has been linked to detecting novelty and saliency in the environment. Dopamine has been, of course, very well studied in its fundamental role in modulating reward prediction, wanting, motivation, right? So some years ago, we wrote a paper to try and understand how much of this work, how much of this fundamental neuroscience knowledge has actually been translated to their artificial counterparts. What do we know about implementations of neuromodulatory signaling in the real brain as against artificial neural networks? So to the left here is a vast landscape of the myriad spatiotemporal actions of neuromodulators, starting from milliseconds all the way to months, starting from nanometers all the way to centimeters, spatial, temporal, right? And these neuromodulators are doing many different things, from ion channels to neuron morphologies to microcircuits to macrocircuits to mesocircuits. But very little of this has actually been realized or implemented in artificial neural networks. So some pertinent questions, that we could try and ask by building a link between neuromodulators and artificial neural networks is, how could neuromodulatory interactions be implemented in artificial neural networks? We heard about crosstalk in the previous talk. And neuromodulators don't function in isolation. They always interact. So there's constant crosstalk between neuromodulators. How can we implement these in artificial neural networks? And how could we evaluate the emergence of synergistic behavioral effects? And could multi-scale implementations of parameter updates and projection-specific functions of neuromodulators, how can they enable artificial neural networks to efficiently reconfigure network-level parameters on the fly? And how could artificial neural networks incorporate sleep-like states, for example, that the biological brain does? We all sleep quite a lot, to mimic biological replay mechanisms and help prevent this so-called problem of catastrophic forgetting in neural networks. So there are many emerging ANN architectures out there that are inspired by biological principles. For example, there are architectures that try and incorporate bursting mechanisms, a burst firing of neurons found in biological neural networks. So there are co-inhibition excitation networks. There are networks that incorporate so-called Dale's principle. There are networks with astrocytes. You're probably going to listen to this talk later today. And there are other emerging architectures like transformers or neural operators or progressive neural network architectures, right? So we started by understanding what kind of neural network architecture is used for what kind of task processing, the implementations. So this is a landscape of different tasks that are implemented by different learning algorithms. For example, by neural networks that use synaptic plasticity, all the way to gradient-based learning, to evolutionary algorithms, to reinforcement learning-based models. And what we see is that there are specific architectures that are mapped to specific tasks. And some of these tasks have not even been implemented despite whatever architectures are out there. But what we found is that almost all of these neural network architectures have no concept of neural diversity, which is a hallmark of the brain, right? So indeed, no two neurons are alike, no two neurons are identical in the brain, and there's a lot of diversity. So there are pyramidal cells, there are basket cells, there are Martinotti cells, there are all kinds of neuron types in the brain, and artificial neural networks don't have cellular diversity like in biological neural networks. So we're developing a so-called dual framework approach to study learning in neural networks. So on the one hand, we try and build detailed models of cortical circuits to try and mimic their laminar organization, starting from superficial layers of the cortex all the way to deep layers of the cortex, trying to model or build biophysically detailed models of cell types and implement neuromodulatory signaling mechanisms. And then we use these detailed models as a reference to systematically simplify and to build a simplified neural network architecture that has neural populations arranged across layers that mimic cortical layers and that have these recurrent corrections going all around. So as a first step of trying to understand what adding cellular heterogeneity to neural network buys us, we implemented a very simple reinforcement learning task. So where the task is to reinforce firing of two neurons via delayed reward, and we modeled neurons based on Eugene Izhikevic's firing types, a celebrated paper back in 2007. And the network architecture we used was a simple recurrent neural network. So about 1,000 neurons with 100 connections per neuron, roughly 4:1 excitatory to inhibitory ratio. We have different spiking populations. And the way learning is implemented in this network is by using a dopamine-based STDP learning. So we learned about STDP in the previous talk. So there's an interesting anecdote about STDP. So this plot that was shown in the previous talk, so the phenomenon of STDP was actually discovered by my PhD advisor, Henry Markram, who also started the Blue Brain project. So that's quite an interesting anecdote. Anyway, so using this model, then we built paradigms of learning where a reward is delivered with a delay of about 1 to 3 seconds whenever pre- and postsynaptic spikes coincide within a 20 millisecond window. The typical STDP protocol. So pre fires before post, the connection is strengthened. Post fires before pre, the connection is weakened. And the reward triggers dopamine release, which enhances the synaptic strength of the connection through DA-STDP learning. So dopamine, the way it was implemented in our network, so whenever there's an STDP event, so dopamine kicks in and strengthens the synaptic connection through DA-STDP-based learning. Just to take a step back, I spoke a lot about morphological diversity of neurons. So this is a periodic table-like arrangement of the diversity of cortical neurons in this little piece of rat cortical tissue that I spoke about earlier. So 0.3 millimeter cube of cortical tissue comprises 55 different morphological types. So, these morphologies are quite complex as flora found in the Amazonia. So you have neurons with axons and dendrites of all shapes and sizes. So it's quite complex, right? And to make things more complex, there's also a mind-boggling electrical diversity of cortical neurons. So there are neurons that are accommodating, there are neurons that are stuttering, neurons that are bursting, neurons that are fast spiking. So there's a huge diversity, a huge morpho-electrical diversity of neurons, even in a very small part of the brain. So how do we try and get a hold of all this complexity, right? So we started by keeping things simple, and we introduced 4 different spiking types in our reinforcement neural network model: regular spiking neurons, burst spiking neurons, fast spiking neurons, and accommodating neurons. Our goal was to try and understand the impact of inhibitory spiking types on learning, because inhibitory neurons have a far more diversity of firing types than excitatory neurons. So we modeled the effect of fast spiking inhibitory neurons to enhance learning of the reinforced synapse, and accommodating inhibitory neurons to impair learning of the more reinforced synapse. So this is a plot of how synaptic strength changes as a function of time. So in red is, of course, the synaptic strength between excitatory neurons. It's blue between regular spiking, between inhibitory regular spiking and excitatory regular spiking neurons. And this is between inhibitory fast spiking and excitatory regular spiking neurons, right? So the learning rate is highest between inhibitory regular spiking and excitatory regular spiking neurons. And of course, we tried to explore this more by trying to include different proportions of spiking heterogeneity to see what impact it has on learning at the synaptic level. And what we found is that a combination of 70% fast spiking and 30% accommodating inhibitory neurons exhibited the best combination for learning enhancement, right? So, and these different combinations are all depicted here. So the model of this slide is that incorporating spiking heterogeneity provides an additional layer for fine-tuning learning in neural systems. So what we find is that having a neural network where all neurons are exactly the same compared to a neural network where there's a heterogeneity of populations, for example, by their firing properties, actually enhances the learning rate by providing an additional layer for fine-tuning. So we also studied the effects of bursting activity in the network, where we introduced populations of neurons with 80% regular spiking and 20% bursting, mostly in pyramidal cells or excitatory neurons. And bursting activity increases network instability when pre and post neurons are regular spiking, but bursting activity prompts unstable learning when pre and post neurons are actually bursting. So what we see here is how the synaptic strength, between pre and post regular spiking neurons is actually quite stable with regular spiking neurons, but is kind of unstable with bursting neurons. As you see here, this learning, although reaches peak right away, it's quite unstable with bursting neurons. So what are the effects of burst spiking at the synaptic level? So what we find is that bursting activity increases pre-post correlated spikes within the 20 milliseconds window, within the spike timing dependent plasticity window. So more dopamine is released, and therefore the synapse is enhanced more rapidly. So this is the scenario, the learning scenario in a network that has only regular spiking neurons. And this is a scenario in a network that has only burst spiking neurons. So clearly, introducing bursting is enhancing synaptic strength in the network as against a network that only has regular spiking neurons. So we then moved a step further where we considered all excitatory neurons as bursting. And bursting activity seems to promote learning by enhancing flexibility. However, compared with the scenario with just regular spiking excitatory neurons, it's quite unstable, right? So regular spiking neurons provide stability to the network, but bursting neurons, position the network as though it were on steroids, for example. So the network tries to do interesting things, but it's very unstable. It oscillates in these epochs of like rapid stability and instability. so what we propose is that neuromodulators can act as switches on spiking activity, helping to balance fast learning and stability. So dopamine drives learning, noradrenaline enhances fast learning by enhancing bursting spiking, and acetylcholine promotes stability through promoting regular spiking, right? So what we see here, for example, is introducing noradrenaline into the network, promotes bursting activity, but then again, it also increases instability in the network. But acetylcholine, on the other hand, tries to promote stability in the network by enhancing the spiking of regular spiking neurons. So this is more of like a crest and trough plot for what dopamine, noradrenaline, and acetylcholine could be doing in a network. So what dopamine is trying to do is to, of course, balance the network, but noradrenaline is kind of pushing the network deeper into a valley. But what acetylcholine is doing is kind of expanding the network state for dopamine to come in and do whatever it's supposed to enhance reward learning. So modulating bursting activity to regular spiking activity could help control this instability in the network, preserving the network state after learning and enabling both fast learning and long-term stability. And neuromodulators enhance fast learning and stability by controlling spiking activity at the synaptic level. So what are key takeaways of this exploratory study? So incorporating spiking heterogeneity provides an additional layer for fine-tuning in neural systems, and neuromodulators can help enhance fast learning and stability by controlling spiking activity at the synaptic level. So yeah, I'd like to thank all my collaborators, especially my PhD student Alejandro, who did all this work, all my funders. So we are a lab in the beautiful northeast of England. Newcastle is a very enterprising city in England. So it has a lot of history. In fact, the railways started at Newcastle. So Newcastle owes the world the railways and the steam engine. And finally, some advertisement. So we have an ongoing call for papers in Ebro Neuroscience Reports, especially from early career researchers, so PhD students, postdocs. So we have a spotlight on scientific advances in systems and computational neuroscience. So I'm the chief guest editor of this volume. So submissions are open until the end of February 2025. So you're all encouraged to submit. So with that said, thank you very much for your attention, and I'm open to questions.
Well, thank you, Srikanth. That was amazing, and it actually very nicely connects to what we were discussing yesterday. I'm so glad we had that lecture by Max, which allows me actually also to follow this much better. We will open for questions. I see Max jumping in immediately.
Thank you very much. This is really fascinating, interesting talk. I have some comment possibly later about this analogy between spiking neural networks that we call spiking neural networks and what we call artificial neural networks, like a generic term. It looks a bit stretchy because they use Rosenblatt model of neuron, which doesn't have neuromodulation at all, like right. Zero. And you use Izhikevich, which different. So my question is, why it is so like concentrated by cortex and cortical structures, practically having out of the scope all the subcortical areas which produces noradrenaline, dopamine, serotonin that you have referenced, and which usually has more complex architecture and more complex functions, including, for example, forgetting or synaptic homeostasis, like in hippocampus. Why it is so concentrated only in the cortex, though the cortex maintains only like 3 to 5 minutes of memory by itself and not capable of maintaining more than that. Thank you very much.
Yes, that's an interesting question. So the primary reason is, of course, the unfortunate strategic location of the cortex in the mammalian brain. So the cortex is the outermost structure. So obviously, it's much more easier to access. And it's also the largest structure in the mammalian brain. So it commands a huge real estate in the mammalian brain. So it makes it easier to access the cortex. On the other hand, we have the tools and the technologies in place now to try and access subcortical structures. But yes, as you said, these are very complex. And their organization is not as straightforward as the cortex. So they do not have a laminar organization. And they have these very huge long-range axons, right, that are really centimeters long, that project from these deep subcortical regions all the way to the cortex. So trying to map all these axonal arborizations is quite challenging as well. So we were largely limited by technology, but I think we have the tools and technologies in place to also try and systematically understand how subcortical structures are organized. And in fact, I'm very interested in studying a brain region called the hypothalamus, which has a lot of different neuromodulators, like histamine, orexin, all these neuromodulators, right? People joke that the hypothalamus is actually the real brain, and the rest of the brain is something that protects the hypothalamus. So indeed, yes, I think the subcortical region is mostly uncharted, and there are lots of discoveries to be made there. So hopefully all this will happen sooner than later. Yeah.
Anybody else? Questions? Well, you can also reach out to the speakers in the breaks.
Absolutely.
Obviously. Well, thank you very much for that. Our next speaker.
Thank you.
And I'd like also to point out that this is the second call for papers by an editor of a journal for our young researchers in the crowd. Hint, hint, nudge, nudge. There's a coffee break afterwards.
Oh, and there's also a little perk. So we could consider APC waivers on a case-by-case basis. And your position is strengthened if you are an early career researcher.
There we go.